Okay. Também.
Good morning, everyone. My name is Diogo Ingracia, and I'm a team leader of the scientific department of the 8th Bioengineering Week. First of all, I would like to thank you for attending our event today, and I hope you find it as interesting as I hope it to be. With that said, I present to you the It's Nano or Never, the impact of nanotechnologies in the healthcare future, which will be moderated by Professor João Pedro Conde. I'm sorry. Regarding the biocoins, please do not forget to scan the QR code. Do not use the SBE app to scan the QR code. Use your camera, please, because we have a problem in the SBE app. And but do not forget to scan the QR code so that you can earn the corresponding biocoins of this session and win one of the 3 prizes at the end of the week. Also, at the end of the 2 presentations, we will have a Q&A moment, so I invite you all to submit your questions through the corresponding section of the SBE app, and let me remind you that by doing so, you also earn biocoins. Additionally, at the end, we will randomly select one of you to receive a small prize that we have prepared, so stay tuned. And with that, I hope that you enjoyed this session, and I hand the floor to Professor João.
Okay. Hello, everybody. So first, thank you for inviting me to be here today. Very nice. We have today 2 very distinguished speakers. So if you look at your program, we were expected to have 3 speakers, but unfortunately, Dr. Lídia Gonçalves has some health problem, right? And she cannot attend. But, we have more time with these two speakers, which is good too. So, I'm going to introduce the first speaker. So Professor Ana Granja, which is over there. So she's a professor at the University of Algarve at the Faculty of Sciences and Technology. Ana obtained her PhD in pharmaceutical technology from the University of Santiago de Compostela, and she's now a senior researcher at the Center for Marine Sciences of the University of Algarve, and she's the leader of the drug delivery laboratory. And I'm sure she will explain us, but her work focuses on inhalation drug delivery strategies, and also, which I find particularly interesting, exploring the potential of natural materials. So thank you, Ana, for coming, and you can start when you want.
Thank you very much, Professor João Pond, and I would like to thank the organizers. Very nice organization so far. I'm sure we will see that through the whole week. It was very nice to see our students engage with this organization tasks, which are very important in your path. So every time you can do it, just join your colleagues for this kind of organization and the organization of these events. Well, that said, I was asked to tell you a little bit about my journey in the academic field and also in research. I confess I felt a little hold. Oh my God, people are already asking me to tell about my experience, but it will be a very nice moment, and it's actually a pleasure. So I entitled my talk, this explanation of my experience of how did particles come to my life, because I realized that particles are in fact part of my life, and they have always been present in some way since the moment I engaged in my PhD. So just to summarize, I have three main steps that I want to share with you. I started to do my graduation in pharmacy in Porto, which finished in 2001. Then I went to Santiago de Compostela to do my PhD, which finished in 2007. And in that same year, I went to the University of Algarve, where I am until these days. So when I did my bachelor in pharmacy in Porto, my idea actually was to work in pharmacy, in community pharmacy. Very quickly, I changed my mind because I realized that I would prefer a lot more to continue my studies and to dedicate my life to research. In the end of the graduation, I managed to have a place to work in an hospital in Porto, but already with my mind in somewhere else. I wanted to go abroad. I didn't want to stay in Portugal. I wanted to have an experience in some other country, kind of close, but not in Portugal, to have other experiences. So during my stage at the hospital, as I had a permanent position in there, I could ask for an unpaid leave, and I managed to go to Santiago de Compostela, where I initiated my PhD in 2002, but still with some secure place somewhere in Portugal, which I could come back to if something went wrong. Fortunately, nothing went wrong. So I started in 2002 in November, and I stayed in there until January 2007. When I finished, I integrated the group of Professor Maria José Alonso, who is a very well-known scientist in the field of nanomedicine. They started several projects in the 90s devoted to the development of nanoparticles, always with an application in medicine. So my PhD was in pharmaceutical technology. So we are the guys that developed the medicines in a program, a PhD program called Development of Medicines. And my particular subject was as the group was developing nanoparticles, they were working strongly at that time in gases and nanoparticles, and they were working in several routes of delivery already, oral obviously, but then also nasal and ocular. And when I came, the challenge that they posed to me was to use these nanoparticles for lung delivery. And I took the challenge, and finally my thesis ended up on a theme that was the microencapsulation of nanoparticles. I will explain later why we need to microencapsulate the nanoparticles, but microencapsulation of nanoparticles for lung drug delivery. In the first year of my PhD, I didn't get a grant from our funding body, the FCT, but I managed to get it for the second year, so I didn't give up. I maintained the faith that I would get it, and I could after the second year. And then I finished the thesis in 2007. In the meanwhile, I had a research stage at the King's College in London for 4 months to work with cells. It was very interesting because in Santiago, we didn't have work with cells at that time. Now they have, but not when I was there. And I managed to learn how to work with cells, which is a very important part of my work now too. So in 2005, the first paper came up, which was a very joyful moment. Obviously, there's always the moment of the first paper for any PhD is kind of a big party. And after my research stage at the King's College, we also managed to have a paper. And when I finished, I had 6, so I was quite happy with that. This is my PhD thesis. As I said, it ended in 2007. And as I said also in the beginning, the idea was to deliver the nanoparticles to the lung, which causes a lot of difficulties because nanoparticles are very, very small, as you know. And when we breathe the nanoparticles, they don't have enough size or mass to deposit during the inhalation time that we have. So when we exhale, the nanoparticles are just coming out, are being exhaled with the air. And we wanted to use these nanoparticles as protein carriers. So we had to develop a strategy to manage to deliver the nanoparticles to the lung in order to comply with the aerodynamic limitations that are part of this drug delivery to the lung thing. So our strategy, there are several strategies. We can form aggregates, for instance, but what we decided to do was a second process of processing of the nanoparticles. So we microencapsulated the nanoparticles using a technique of spray drying. And we managed in this way to have carriers with suitable aerodynamic characteristics to reach the lung. And once there, they can just dissolve the microparticles and release the nanoparticles in the desired place. So after finishing my PhD, I really wanted to come back to Portugal. I'm from Braga, very close to Famalicão, which is quite close to Santiago de Compostela. So I was exchanging the country every week during 4 years and something. But anyway, I have decided that I wanted to give back to the country what the country gave to me with my PhD grant. So it was decided very soon that afterwards I wanted to come back to Portugal, and I started to see the opportunities, and I found a place to stay in the University of Minho. So I initiated my postdoc in the 3Ps group in February, finishing the thesis in January. In February, I started in there. But very soon, I had the opportunity to apply for a place in the University of Algarve. So in the same year, I just started my placement in the University of Algarve, where I integrate the teaching staff of the Integrated Master in Pharmaceutical Sciences. I teach in pharmaceutical technology. At that time, the graduation in pharmaceutical sciences was a new graduation in the university, so they were in need of people of all areas, basically, and also of pharmaceutical technology. So I just went there, and I'm staying until today, very happily, because it's a very nice place to live. And in 2008, I started to see what I could do to continue my research, because when one starts to teach, oh my God, it's so a new world with a lot of things to prepare and a lot of things to read. So it was quite a difficult first year. But after a while, I got the time to start thinking about research and what I'm going to do. The University of Algarve is quite a small university with many different areas of knowledge. So I integrated at that time the Center for Molecular and Structural Biomedicine, which doesn't exist anymore today. It was part of the IBB, which is also from the Instituto Superior Técnico. And in this way, I could contact a little bit with the biomedicine field, which was the closest I had to the pharmaceutical field, because in the Algarve at that time, there was nothing on pharmaceutical technology. And we needed to develop new things, which was a very interesting challenge, because I had to start from zero, not only in the classes, but also in research. And obviously, it started with asking for funding, and we managed to have our first funded project in 2010, which was a project very well fitting the background that I had. It had to be that way, because I didn't have any other support. So I needed to do something that I was familiar with. So I had a project on the development of nanoparticles for the nasal and lung delivery of peptides and proteins. And this is how our group started in 2010. And through the time, I did two major decisions. One was that all our activity, or practically all the activity, would focus on pulmonary applications. And another one, because we need to engage with the needs of the region. So I needed to understand what Algarve was needing in relation with this field of biomedicine and pharmacy. And so I also decided that I needed to explore natural materials, potentially local materials, as potential excipients to use in the pharmaceutical field. So we mainly work with polysaccharides, and I have in there some of them. How would I like locust bean gum? I imagine most of you know it is alpha-hoba, very known from the Algarve region. It is a lot used in cooking, but also in the pharmaceutical industry. So with using several of these materials, we have developed another research line and another project, very interesting project that we developed in this field of inhalation, because we proposed an inhalable tuberculosis therapy. Tuberculosis is an infectious disease that is a respiratory disease. It appears when an infected individual just expels droplets with bacteria, and another individual that is a healthy individual just inhales this bacteria, and the bacteria will, if we cannot manage and eliminate the bacteria, they will be hosted in the alveolar macrophages that are inside the lung. So the problem is that we have an adequate therapy for tuberculosis for many years, from the 50s, but the problem is that it is a therapy that is too long, at least 6 months, 9 months. It's very common, even more, and it comprises 4 antibiotics for the first 2 months, and then 2 of the antibiotics for the rest of the time. It's a lot of tablets to take. It has strong side effects. So in many cases, people are not really complying with the therapy. We are having resistances. So it's a disease difficult to treat in some cases. So we just thought that as it is a respiratory disease, it would be adequate to propose an inhalable therapy that can directly deliver the antibiotics to the place where they are needed. So we developed this strategy of incorporating the antibiotics that are in the current clinical use inside microparticles that we endow with the adequate aerodynamic characteristics to reach the alveolar zone. But not only that, we also wanted to provide a strategy that could benefit, let's say, from the specific properties we identified in the host cells. The host cells of the bacteria, as I said, are the alveolar macrophages, and alveolar macrophages located right down there in the alveolar zone are cells that express some specific receptors. Okay, so we identified some of these receptors, and we realized that some of the polysaccharides we have been working with have units, chemical units, that can target specifically these surface receptors. And we thought, why not? Just join all the knowledge we have and to propose some microparticles that could provide a potential benefit for and benefiting from this targeting strategy of the alveolar macrophages. So it went very well. We just finished this project like 2 years ago. We don't have funding for more. It's always about funding, as I think you know. But we have managed to have a patent on these outcomes, these particles that was conceived last year. I still have time, don't I? One of the things that we have learned very strongly was that some of these polysaccharides, in fact, have very interesting properties. And we identified, because of that project on tuberculosis, that some of them are really good at targeting the macrophages, which are phagocytic cells. So what we thought is that we could benefit from those properties also in the field of vaccine, of immunization and vaccine development. So we are working currently in some strategies that include the development of inhalable vaccines, mainly targeting respiratory diseases, naturally. So we use some of these polysaccharides to produce microparticles that just microencapsulate antigens. So we hope that the microparticles will provide a more intimate contact with the phagocytic cells, namely the dendritic cells that are the main APCs. So we will be able to have not only a systemic immune response through the production of immunoglobulin G, but also the production of secretory immunoglobulin A, which is responsible for the local immune response. And in this way, we hope that we can protect the lung further, comparing with the conventional delivery of vaccines to the parenteral route, which implicates an injection. So we proposed this project to the Portuguese Society of Pneumology, and we could be awarded with one of their prizes. So with this money, we are just initiating the project. We have a PhD student, dedicated to this line in the second year of the PhD right now. One of the other things we also identified, that when we work, I think that everyone experiences this. When we work, we identify a lot of limitations. And I think that one of the goals that we should have is that when we identify limitations, we should try to produce whatever it is that allows us to overcome the limitations. So one of the limitations that I've been identifying for years in my field is that I work with microparticles. Microparticles are powders, are dry powders. We work with dry powders only, because in inhalation we can have liquid systems, also powdered systems, and we always work with spray drying, so ending up with powdered systems. So it's a solid. When we inhale these solids, the solids are dispersed through the whole lung, and the lung is devoid of liquid. I think you all know that. We don't have liquid. We have mucus in some parts of the lung, in the upper parts. In the alveolar zone, there is no mucus at all. It has just a small amount, a very thin layer of a lining fluid that prevents the lung to collapse when we are breathing, okay? So can you imagine how is the release of the drugs from these systems that we are delivering to the lung. It's difficult. It's much more difficult than when we compare to the oral route or even the nasal, where we have more mucus. Okay, so not having liquid, it's really a limitation. And it is also a limitation to try to mimic these conditions when we are doing in vitro studies. In my group, we always go from the development of the systems, we characterize exhaustively the systems, and then we go ahead to biological assays. And obviously, we start with cells. In later stages, if the results are very good, we can reach in vivo studies, but it's not frequent. Typically, we end up with cells. Putting these dry powders in contact with cells, it's very difficult, because typically people, what they do when they work with particles and they want to expose cells to the systems, they just mix the systems with cell culture medium, the medium that the cells need to survive, and they just do the contact in this way, because in most of the delivery routes, there is liquid. So we are mimicking the conditions when we do so. But that's not the case of the lung. So how do we do that? How do we disperse the powders over the cells? There are no equipments to do that. Well, there are some very expensive, which we cannot afford at all, but which are not exactly used for that, but can be adapted. Okay, but if we want to get a very small equipment that just enables us to do something as simple as dispersing a powder over cells in culture, we don't have that. So I decided some years ago that we needed to do something regarding that. And my idea was that we could just have our cells in a support, imagine a plate, for instance, or a petri dish, something that is adequate for cell culture. And then we should have a device that we could use to pump air and disperse the powders over the cells, so that the cells would be in contact with the dry powders in the absence of liquid. They still have a very small amount of liquid, because without liquid, they just get dry and die. But the liquid is very, very small amount. It's not enough to disperse the particles in the liquid and expose, as it doesn't mimic the real situation. So we just, after several years dedicating to this, we finally managed to develop this device that you see here, and it became true 2 years ago or something, with a PhD student that we had. So we now have one device that can help us to, as you see here, this is the aspect of the petri dish after the powder was aerosolized on top of the cell layer. So this is also a project that we managed to initiate a patent process. We only have the submission to the EPO, but we are patent pending, actually, and we hope we can succeed with that. And I think that's it. Thank you very much. I hope you enjoyed. I thank you all the funding that we have been having, and obviously the very helpful collaborations. Without collaborations, there is no research at all. Thank you.
Thank you, Anna. So this was very interesting, and I just wanted to comment to thank Anna for sharing the trajectory, her trajectory with us. So some of you, we are mostly second cycle students here, I think. So most of you, many of you will want to do a research career, and it's a bit like what Anna said, that you have to do a PhD, do a postdoc, become unique, go somewhere and find an empty space and have to build a group. And these are all big challenges, but of course, also very exhilarating when you succeed. And most of the people are smart enough to succeed. So thanks, Anna, again. You'll be able to ask her questions later. So now, the next speaker is the curiously named João Conde. So Professor João Conde is at this moment also a research group leader, and he will explain us a bit about this. But he's a professor at Medical School of the Universidade Nova. So João also has an interesting trajectory. So he received his PhD in biology at the Universidade Nova, I think, with Pedro Batista, and also in collaboration with the University of Saragossa. And then he did a postdoc in the United States at MIT, actually at the Harvard MIT Division for Health Science and Technology, and he was also in London. Then he came back to Portugal. So we all do the same mistakes, I'm afraid. So he came back to Portugal, and he was a junior researcher at IMM. IMM is the research institute of the Faculty of Medicine at the University of Lisbon. In 2019, he won a big research grant called the European Research Council Starting Grant. In 2020, he moved to the Universidade Nova as a professor. And in the meantime, he had other adventures, namely starting a company, etc. So, João, on behalf of the organization, thank you very much for coming here to share with us your experience.
Thank you so much. Thank you so much for the introduction. And thank you so much for the organizing committee and for the invitation. It's always a pleasure to talk about what I love to do, so it's very easy. And today I'm going to talk a little bit about my path on this precision nanomedicine. It's what I've been doing in these last 12, 14 years. And then I will show you a little bit what the things that inspired me to work on this field over these years. So, like Professor João Conte said, my path began at least with a PhD. I won an FCT grant to do a PhD in both FCT in Caparica and Universidade Nova and Universidade de Saragossa. So I won like this grant to go abroad for 1 to 2 years and do half of the project here and half of the project at the University of Saragossa. And one big consortium, which was a 5-country consortium that was called nanotech. That had groups from Italy, from Glasgow, from Germany, from Portugal, and from Spain. And so we were responsible, the groups in Portugal and in Spain were responsible to do the nanomaterials, the nanoparticles, the functionalization with different biomolecules. And then all the other groups were supposed to test it in these biological systems, in vitro and in vivo. And so it was an amazing experience for me because I could engage in this great collaborative consortium in order to build multifunctional nanoparticles for gene silencing to use in gene therapies in cancer. And then after finishing my PhD, I applied for a Marie Curie fellowship where I went to go to the MIT. But just to tell you a little bit, before this, I contacted like 15, 20 groups at the MIT because I knew that I would like to go there because what the groups were, I think, the most fascinating me, the things that the work that they were developing. And so I contacted like 15, 20 groups and only one replied. Which was the one that I went. And which the group from Professor Natalie Artsy, she is now at Harvard Medical School. And she asked me, you need to come right away. But I was 6 months from finishing my PhD. So I said, now how I'm going to, what I'm going to do, because I really like to work with her. And so I applied the Marie Curie. That's why I applied the Marie Curie, nothing more. I just applied the Marie Curie to go to this group. So, and it was because of that I went. So I won the Marie Curie, and so I flew to Boston to the MIT, and it was an amazing experience. Probably the best experience of my life, the best experience of my professional and also of my personal life. My second son was born in the US, so it was really an adventure. So it was really coming out of my comfort zone, and both professionally and personally. So it was really an amazing experience. Then the third year of my Marie Curie was to come back to Europe. So I came back to the Queen Mary University of London to do the third year of the grant. And then when I was at the Queen Mary University, things didn't go very well. So I just was at this time that I started writing my ERC. Because I knew that I would like to come to Portugal. I don't know why, but I don't know. I think we have this idea of, I don't know, this saudosismo. So we always want to come back to our roots. I don't know. And so I knew that I wanted to have my group, and I knew that I had to apply to a big funding. Otherwise, would be very difficult. I don't know why, but I'm sorry that it has to be like this, but most of the times it is. And so I started writing the ERC when I was at the Queen Mary, and then I wanted to come back to Portugal, so I contacted several institutes if they wanted to host me as an ERC grantee. And also only one replied. And so, but anyway, so then I won the ERC, and then now I have my group since 2020 at Nova Medical School. I'm very happy there. I think it's a very good environment. It has these. We are very close to the clinics and to the clinical research. And so I think it's really relevant for what I've been doing. So this is my path. I had the lucky to work with amazing people. So I was really lucky to have all these experiences. But I always took a lot of risks. So this I think it's important for you. Come out of your comfort zone, take risks, because it's where you are going to learn more. It's by taking risks. So that's for sure. And to do new things, inspiring things, and important things and relevant things to the clinic. So this is really important. So just to tell you a little bit about how it started, this idea of this intersection between nanotechnology and oncology. It has been all my life. I've been developing systems to treat cancer or to diagnose cancer also. I started by developing kind of sensors and colorimetric assays to detect cancer also. And so you all know that cancer is probably one of the biggest burdens that we have. So, and now I'm taking part of this consortium. It's one of the biggest consortiums in the world in terms of global burden disease, from the Institute of Health Metrics and Evaluation. And so we all know that cancer, it's the biggest burdens that we have. It's the second cause of death worldwide. And so we really need to develop new systems, and we are actually in urgent need to reshape the way we treat cancer. Because I think that the clinics and the clinicians and our system is really addictive to the way we always delivering the drugs that for cancer patients. We are always delivering the same way the drugs that we do for 50 years ago. So we really need to shape. And I'm really happy actually that we have here today another group that also works on these localized therapies, because most of the things that we saw to treat cancer is systemic delivery. Like for chemotherapy, that we deliver the drugs intravenously and they go everywhere. and they accumulate everywhere in our body, and just a fraction of that drug goes into the tumor. And so we are really in need of these new delivery systems. And this is all about nanotechnology. And actually, when I first started doing research, was not on nanotechnology. My fellowship, like for the estágio, I don't know how you say it, for my bachelor, I did it for breast cancer. It was an epidemiology studying different genetic variants for breast cancer. And so only at the time I was introduced to this idea of nanotechnology, and if you see, this is all about delivery. And it was with this essay and this meeting from Richard Feynman that really inspired me to start in this area. And it was when I read this essay from Richard, that there's plenty of room at the bottom, that I realized that nanotechnology was really it has an enormous potential to change the way we treat diseases and we diagnose diseases. And so this idea with Richard, just for you to know, it started like in 1959, this time, where computers were the size of these entire rooms. So to say that, and Richard said a lot of things that were really inspired, like why couldn't we write the whole volumes of an encyclopedia on a head of a pin? So this was really a revolution at the time. Another thing that he also introduced was this idea of swallowing the doctor. So kind of the systems that we are developing now, actually. And he said, why can't we have like a sort of robot, a nanorobot that could enter our body and chase the diseased cells and fix them? So this was in 1959. And so I was really inspired when I read this essay, and I said, how was it possible that so many decades ago, this scientist, which was a physicist, really had this vision. And the truth is that many decades ago, it was only a vision. But now it's really a reality, because if you think about the nanoparticles that we have been developing for tumors, so we can actually functionalize all these carriers with all types of molecules, from proteins to nucleic acids to vitamins, carbohydrates, anything. And we can use them to modulate genes, or to modulate the immune system, or the immune cells to work for the tumor, and to put them work against the tumor. And so we can actually engineer all of these carriers to be responsive to stimuli, to internal stimuli like pH or hypoxia, or external stimuli like light, for example. And we actually develop a lot of systems also with lights that are responsible to light, to use as a photothermal therapy, for example. And so this is it has a huge potential in terms of therapy. And so we can actually develop any kind of particles to use in the tumors. And so when I look at the structure of these carriers, they are really amazing, because you can actually add whatever you want in the surface of these carriers, and also inside them. So you can encapsulate any therapeutic molecule. They can be built of all types of materials. It can be inorganic materials, organic materials, and they can even be covered with cell membranes from cells, from red blood cells, from immune cells, from cancer cells, in order to camouflage these particles when they are entering our body. And so this really fascinated me, and we use this for many years to treat cancer. We also, and that's what I wanted to talk. The first experience that I had with nanotechnology was actually from the diagnostics part, not only on the therapy. So I first developed systems like this colorimetric assay with Professor Pedro Batista, where we could build these two-color like negative for we were detecting some sequencing from some sequence that was overexpressed in a certain type of tumor, for example. And so we could use these sensors to develop a negative positive assay, like the colorimetric assays to detect DNA, RNA. And so this was the first part of my PhD also, and to detect and inhibit the expression of a specific gene. But there's a lot of other types of systems that can be used to detect, and we have this with COVID-19. Actually, I think taking your motto like it's nano or never, I think nano it's now more than ever a reality, because if you look at the COVID-19, this was a really revolution in terms of the nanotechnology, because if there was any skepticism in using materials or nanomaterials in humans, that skepticism completely vanished. Actually, we saw this type of, for example, the diagnostic device like the rapid antigen tests, most of them were made with gold nanoparticles that have an antibody that binds to the viral antigen of SARS-CoV-2. So there's a lot of devices that for diagnosis with COVID-19. But the most striking example from the COVID-19 was the use of the messenger RNA vaccines, and two of the messenger RNA vaccines, the ones that were developed from Pfizer and from Moderna, they were made of lipid nanoparticles that were carrying the messenger RNA against SARS-CoV-2. And so, if I may ask here in the audience, can you put your arms up if you were injected with the Moderna or the Pfizer vaccine? So can you look around? This is the power of nanotechnology, because all of you were injected with lipid nanoparticles carrying a messenger RNA intramuscularly. So this was really a revolution. And then we published just now this paper where we can see here from blue and from orange, you can see the distribution of the nano vaccines in the world, the messenger RNA vaccines from Pfizer and from Moderna, and you see that they were inoculated in hundreds of millions of people. And so this really shows the power of nanotechnology, and that is everywhere. And now it's a reality. Actually, I think it's a great time for nanotechnology. What we are seeing this now. And so one thing that I would like to show you also is that these technologies take time also to mature, because if you look at this was something that people was always talking, these nano vaccines, they are really new, they are based in very recent technology, but it's not true. If you look at since the discovery of messenger RNA in 1961, the first nanoparticle that delivered this kind of molecule was built in 1976, and it was done by Robert Langer at the MIT. And so it was since 1976 that, and also in 1978, that the first messenger RNA lipid nanoparticle, very similar to the ones that we got inoculated, started like in 1978. And so it took from 1978 to 2020 to have these messenger RNA vaccines. And we will see in the future a big boom in not only in oncology, but also in other diseases like genetic diseases or infectious diseases, not only for COVID, but also for HIV, Zika virus, influenza, and also for cardiology. So there's a lot of companies like Pfizer and BioNTech, but also Moderna and other companies that are developing these messenger RNA vaccines, most of them made of nanoparticles like this. so I just wanted to show you how this really inspired the work that we have been developing in the last years. This is an example of the landscape that we developed in the last 10, 20 years, last 10 or 15 years. And so we developed a lot of these particles that were made of gold nanoparticles or quantum dots or polymeric or lipid. So a lot of different materials. They could be used for systemic therapies, or they could be embedded on localized biomaterials like hydrogel patches or microneedles. And so, and we have developed this for several cancer types, from lung cancer, also gastric cancer. Actually, we also developed a localized for lung cancer, a little bit like what you saw before with this last presentation, and also for colon cancer, breast cancer. And our main goal was always to tackle some of the most striking biomedical problems in cancer. One of them is the preventing recurrence. As you can see, most of the tumors, they recur and tend to recur when we stop the treatment. And that's actually one of the problems with the systemic therapies, is that we're always in need to give more and more administrations in order to compete with this exponential growth of the tumor. Also, we developed systems to prevent and inhibit metastasis just by controlling the tumor microenvironment, the primary tumor microenvironment, and we could inhibit metastasis from establishing just by targeting the primary tumor. Or, for example, block multi-drug resistance, which is also a big problem because cells acquire resistance when they are administered to some drugs, and so this is really a huge problem, and that we tackled in this last, and that you can see if you are interested to see one of our studies also. And so these are the four main corners of my lab that I have now at Nova Medical School. One of also the biggest problems is the heterogeneity that we see in the tumors. And this is the scope of the ERC project, is to design materials that are able to profile this heterogeneity and to profile the response to therapy, to different therapies in the different cells of the tumor microenvironment. Because if you think about the tumor, this is a very complex system made of different cells, like immune cells, normal cells, tumor cells, and they are all together in this very complex system, and not all of them respond the same way to the same agent and the same drug. And so this is really important to develop these systems that are able to profile in a cell-by-cell basis the heterogeneity across different cancer types and even in the same cancer type. Also developing materials that can block multi-drug resistance, or, for example, develop systems that can be combined in the same device. We can have all types of therapies, and actually this is the beauty of this type of materials. We can incorporate in these materials all types of therapies, so we can have gene therapies, drug delivery, phototherapies, all in the same device, and have a synergistic effect over the tumor. And so this is really important. And also the metastasis and the prevention and the treatment of the metastasis, it's one of the biggest problems, because in some types of cancer, for example, breast cancer, 90% of the deaths are related to metastasis. And so this is actually one of the biggest hurdles in terms of the cancer therapy. And so just to finish a little bit with this path and all of the lines of research that I was inspired with, and I wanted to grow. Because when I started doing the colorimetric assays, I remember perfectly well that I said, I don't want to do this anymore. I just want to do more more about the therapeutics. And this is an advice that I can give you. Try to look for and to be inspired with things that you want to do, to have a vision for the future, more than just to do what everybody is doing. So try to do different things and to be ambitious and bold when you raise scientific questions and when you raise goals to your path. I think this is the most important. And one of those bold things that we did, and actually this is something that I'm really proud, because I think that all of us that are doing materials for therapy, this is what we want, is that our materials can be closer and closer to the clinics. And so we started this company based on the discovery of a new drug for brain cancer, for glioblastoma, which is great for brain cancer. It's the most aggressive brain cancer type, and is one of the most hard to treat, because if you think about our brain, he's protected by one of the most efficient barriers, which is the blood-brain barrier, that does not allow to pass most of the drugs from the rest of our body to the brain. And so there's a lot of therapies, and most of the conventional therapies do not work in brain tumors. So this is really an unmet medical need. And so these all ideas started with the discovery of a new drug, which was the pipalongamine, which is derived from a natural product, from a pepper plant. Actually, it's also a natural product. And we discovered this drug by using an artificial intelligence algorithm. So we found, among a lot of ligands and a lot of biological targets, we found that this drug could target a specific channel that was overexpressed in these brain tumors. But then was the most interesting part of this is that when we tried to deliver this drug systemically, and it could be intravenously, intraperitoneally in the abdominal cavity, it could be subcutaneously, and what we saw is that the drug could never get to the brain. And the tumor growth exponentially over the time. And so that's how we came the idea, why don't we treat these tumors locally? And so instead of having something that goes through our body and accumulates in the liver, spleen, livers, or it has a lifetime of seconds, of minutes in our body and it's cleared out very easily, we developed a gel that could be implanted in our brain without disrupting the brain tissue, but just bypassing this blood-brain barrier, and could diffuse through the gel the drug to the tumor tissue. And so what we saw was that for the systemic therapy, the tumors grow exponentially, and with the local therapy, we could almost completely shut down the tumor cells from the brain tumor. So this was really a revolution at the time in terms of therapeutics, also because, and I think this really brings us the idea that we really need to change the way we view and treat cancer, because I think that perhaps we don't need new drugs we just need new ways to deliver these drugs. And so, because we also tried with temozolomide, which is the drug that is used in clinical trials, in clinics, and we had also very much better results than by injecting the temozolomide systemically, like people get from the chemotherapy. And so this was really this. I think this is an idea that I would like to put put the seed in your head, is that maybe we need new ways to deliver, and nanotechnology is certainly the best way to do this. And so I just like to give you this last idea, is that this work and our work was only possible due to the collaborations that we have in all over the world. So this is a huge teamwork. Never try to do things alone, because you'll not succeed. That's for sure. So this is where you can grow more. You can learn a lot from many other groups that have different experiences. So this is really a huge teamwork, and that's I wanted you never to forget this. And so, last but not least, I would like to thank my group, which is, and I think they are the ones that make things happen. And so I really like. I think it's a huge opportunity for me to work with and to mentor these remarkable young researchers. And so I think this is the best of my job, is to see them grow. And so I think this is definitely the best benefit of my job. So I'd like to thank you, and I'm open for questions.
Thank you very much. Well, thank you for this inspiring talk. Actually, in this semester, they also had a session on personalized medicine and this type of things, and that has also a bridge to what was said today. I also wanted to mention that we as Portuguese, we not only have saudade, but we are also usually very negative, and we have a set of really good scientists everywhere in all our universities. We have good companies. So, I think we need to be a bit optimistic. We have a lot of conditions to be optimistic about our future. So now we have some discussion. And strangely, the organizers decided to do this via the apps to avoid any kind of contact. So let me just see here. We have a set of questions. Let me just see. You scroll. Okay. I'm just scrolling here. So there is one here. Let me. Can I edit this a little bit, just to not say everything in the same order? So there's an interesting one here for both of you. Since the area of nano is a recent area, is it difficult to find articles to help you in your research? Does one of you want to say something about this?
If it is difficult to find papers, articles, no, it is not, for sure.
Every week, thousands.
Thousands. Sometimes I think that the difficulty is to choose the right ones when you don't have enough time, which happens frequently. That might be the challenge. There is always so much information coming up. And that's one of my biggest complaints, because as a teacher, I have a quite high teaching burden, and on top of that, all the management activities that we have. I'm, for instance, at the moment, I'm the director of the graduation in pharmaceutical sciences. So I have a 5-year course to manage with all the things that happen. And then we still have to be active, and we want to be active in research. And it's really difficult to keep us up to date. Okay, so thousands of papers. Some of them probably are not of so good quality. So that's the first thing we need to do, to choose the right papers, and then to find the right information concerning what you are looking for. Because there are a lot of things coming up. Lots of things.
Yeah, and this is actually an important discussion, because there's a lot of things happening. Actually, we are in process of publishing a meta-analysis on the last 20 years of the nanoparticles used for in vivo delivery in cancer. We actually analyzed thousands of papers to extract the information that was being published, and there's a lot of things to improve in terms of characterization of the materials that a lot of people sometimes miss the characterization. And so this is really important also to filter a lot of the results that are coming out. And this is an important thing, is that I know that we are at the time of that pressure to publish, but I think we should wash out of that thing completely, because we don't need to publish more. We just need to publish better and to publish more complete results, more important results than just to publish this and that. And this is something that you will, if you will follow research, and if you get the research, it's important to take in your mind that it's not the most important part is to publish. So I think it's important, also to mention.
Maybe this is more for João, because you have the experience both of some academic research, but also industrial or company research. So somebody asked, so the area of nanotechnologies is an emerging area, especially for research. Is it possible to do research also in companies, or is research in nanotechnologies applied to medicine only at universities? Do you have a comment for this?
It's important. It's possible to do it in companies. Actually, the TARTEX, it's one of those examples. We also do research, and we actually preparing what we did in the lab to prepare to go to the market. There's a lot of different processes that need to be done in terms of production, because in the lab everything can be done in small quantities, and then when you go to a company, you need to have this scale up notion of what you are trying to do, and also because we did all of this in mice, and then it needs to be done in humans. The volume of the brain is completely different. Everything it's different. So there's a lot of companies in the world where you do research. I do think I'm like João said, I'm very optimistic in terms of this area. In terms of we have excellent researchers, and we have more and more companies that are starting now developing these materials, but we need to invest more to create these companies, and the big pharmas need to invest more on these small companies. And in Portugal, there's a lot of most of the big pharmas do invest more on the commercial part and not only on research. But if you see it in many other countries like the US, the big pharma feed themselves with these small companies and small startups that do a lot of research actually in the company. They have labs like we have in academia. So this is possible, and I think it's needed more and more than ever.
Okay, and then you can sell your company and become a billionaire when you are 40. It's good. There's a question here, maybe it's for Anna, which is the following: how did you come up with this idea, for example, of the alpha-ROBA compound? Because, as I mentioned here, in the case of chitosan, this is a common polymer. People have known about this. But how do you choose or select more exotic compounds? Because there are a lot of molecules, right? So, do you have any guidelines for us?
Well, actually, it was kind of a need because when I went to Algarve, we had difficulties in funding, as we have all over the country and in other countries also, but in Portugal we are always saying the same. And we need one very important thing that I learned through the whole journey is that you cannot think that you will do research on what you want. You need at least to make that along with the choosing the right areas that are the focus of funding. And for instance, if you go to Europe, you have in each telling the name now, but from a set of years, for instance, you have the priorities that Union is funding. Okay, so you need to study that and to know what you can do in order to have possibilities of funding. So when I went to the Algarve, I realized that I also needed to find something that the region found attractive, because in some, I didn't show it specifically, but some of the projects we have been having, even if they are quite small, it's always money. and much of the money is devoted to exploring new potential of the proper region. So we are working with materials that are coming from the region. And as you know, alpha is, I like the word exotic. I will start to use it. It's a not well-known material. Like others, we work, if I tell you about fucoidan, for instance, or glucomannan, they are not typical names that we hear about. But if you go to look for some literature, you find some things. When I started to work with alpha-HOBA is already used and approved by our agencies for oral delivery. So it is included in tablets because it can be used as an emulsifier, a stabilizer, etc. But mainly in tablets and only for the oral route. Okay, so it is not approved for lung delivery or any other route of delivery. But when we started the second project that we had, or when we started to develop the idea was on tuberculosis, but benefiting from the knowledge that we could extract from the literature regarding targeting. We wanted to target the cells. And we realized that the macrophages, which are the hosts of the bacteria, have the specific receptors that are, well, they are varied, but some of them have a special affinity for many polysaccharides, because polysaccharides have mannose in most cases, or in many cases, like alpha-HOBA. Alpha-HOBA is a galactomannan. It means that it is a chain of mannose, and then it has lateral chains of galactose. So we have both types of units, and they both have affinity for macrophages, as well as fucose, for instance, from fucoidan. So when we choose the materials, well, we need to have a reason for that, obviously. One of the reasons was because it fitted our objective, but the other reason was because I knew it would be attractive for the region. And we need that because some of the funds of the local funds are enabled by these kind of choices that we do.
Let's not be so cynical. Can I just continue? One question here that I think is for João is somebody did not really understand how you can use these nano carriers to prevent metastasis.
So,
do you have any?
Yeah,
kind of
I can tell you what we did in the past. One of the studies, which was one of the most striking ones, was for breast cancer. So we use a breast cancer model, metastatic breast cancer. And as I told you, 90% of the deaths in breast cancer are related to metastasis. And so what we did is that we analyzed hundreds and thousands of breast cancer samples from clinics. And we found that there were 2 microRNAs that were deregulated in the tumor, in the primary tumor, through biopsies of these patients. And so what we found is that these 2 microRNAs regulate the protein from the cytoskeleton of them. And so this had a huge impact on the invasion of the breast cancer cells. So just by regulating the expression of these 2 microRNAs in the primary tumor, we prevented the metastasis from happening, to go from these cells that were on the primary tumor to migrate to other organs like liver. They also migrate a lot to the lungs and to the brain, even to the brain. And so we could completely eliminate the metastasis to the liver and to the brain, and reduce a lot the metastasis in the lungs for more than 80% reduction, just with one administration of a gel that was implanted on the primary tumor. So this really reflects on this idea of how a local therapy can also have a systemic effect. And the nanovaccines are these, because we are injecting the particles in the intramuscular, right? So we are pressuring those intramuscular cells to produce the spike protein of the SARS-CoV-2. So they produce this viral protein, and in order to our immune system to create this memory against a future infection. And so it's like the vaccine. It's also so it was kind of a this study that we developed was kind of a nano vaccine for breast cancer. So we could, before metastasis establishing, so we could prevent them so these cells to migrate to other organs. So it was really amazing, because it was even more amazing because the therapy that we applied locally didn't have any effect on the primary tumor, but it had a huge effect on the metastasis. So it was really surprising for us at the time. Yeah.
Thanks. I also had not understood this, so this was a good question indeed. So now I have a question that there are two questions here that are about more or less the same thing. So let me formulate it, and then maybe Anna can answer first, and then João. So the version for Anna is: are there any prospects for clinical trials regarding this vaccine by inhalation? And then after, for João, if this gel has already been used in any advanced animals or humans, or if it's only on in cell experiments. Okay. So any clinical trial perspectives?
So I can start. You know that lung delivery is a challenge. It's really difficult because we have many limitations. It's a very protected organ, very sensitive, and I would say that one of the biggest challenges is not really the aerodynamic part, because it's something that is there, and you need to, if you think on lung delivery, you need to comply with aerodynamic requirements. That's why nanotechnology is difficult. We always have to process nano systems again to endow them with the adequate aerodynamic characteristics. But when we do work like I do, using different materials that are not approved, the big challenge is to demonstrate the absence of toxicity. And that's one of the first things we need to do. And one of the things that I am always asked for. Every time I present the work somewhere, there is a question coming in: what about the toxicity of these materials to the lung? So that's something we are trying to contribute to and to generate information regarding that. We didn't manage to have that project funded so far, but we didn't give up, and we produced some results already regarding the use of these polymers, because before thinking on the real efficacy of the system itself, on the therapeutic part, we need to go a little bit before and to think on the possibility to use the materials that we identify to have such a huge potential. So actually, they are not answering directly. There are not any clinical trials in line regarding this, because we need to produce a lot more information, particularly on the toxicity of these materials. You know what? When I did that project of tuberculosis, we protected the work with a patent request before presenting it anywhere. And in the first conference we went to present it, it was myself that went there to explain everything I could. And I chose a very industrial type conference, proposed. And obviously, after the conference, I was contacted by several people. And one of the companies was a Portuguese company, very interested in the patent. But the first question was, what about the toxicity of the materials? So we had several meetings, but in the end, they just gave up on the idea of working with us because of that. So after that, I realized that, okay, I really need to try to do something regarding this, and that's what we are doing currently in the group, in parallel with the other research lines I showed to you.
So next time you have a tart alcohol, when you brief it, we'll have to see if anything happens. João, you want to comment about the gels and the possible...
We are actually at the Targetex, we are preparing those gels to go to clinical trials phase 1 next year. So I hope we are really close to go to that, and I think that's really good. And this is really good that also Anna talked about toxicity, because actually the drug that we are using for when it's delivered systemically, it has a lot of toxicity to the kidneys, for example. But just by encapsulating this drug with a cyclodextrin, because we did a cyclodextrin carrier indirectly into the brain, and we saw that there was no leakage to other organs. We could avoid complete. So the toxicity is also related about this route. The routes of administration plays a key role also in the toxicity part. So and so these ideas. So we are really close to at least to go to clinical trials phase 1. To go to clinical trials phase 2, we will need
a partner.
10 million or 15 million euros. So it's going to be. But one thing that COVID, so I'm really optimistic now, like Sean said, because like what COVID also showed us is that it's possible to approve things faster than we have until now. It's possible. It doesn't have to be like the vaccine for poliomyelitis that took 10 years to go. So we really need, this really shows that the regulation needs to be more, it needs to be faster, I think, especially for this kind of diseases like brain cancer, where patients have 12 months after diagnosis, 12 months survival. So it's really a need to develop new therapies. But I think we are close, at least to be a little bit closer to the clinics, which I think is our main goal always.
There's a question here that I really like, although it was not loved by the participants, because I just realized that this has a ranking of the questions by this one didn't get any hearts, but I think it's quite interesting for a short answer, because then we'll have to wrap up, which is this idea. So our research, actually mine too, is very experimental, right? It's very lab intensive, but in many fields, simulations like in silico studies are a way of actually expediting some of these and having a bit less effort. So I wanted to ask both of you, you can in any order you want, if in your research, is there any role for simulation for this type of in silico studies besides the laboratory experiments?
Actually, we did some in silico studies of this new drug with and the interaction with the target. We saw how was the docking of this drug to the proteins and to which amino acids. Of course, then we corroborated with cryo-EM and crystallography in order to see exactly how this new drug could actually block this channel that was overexpressed in brain tumors. And so we actually benefit a lot from these in silico simulations. And also artificial intelligence is a kind of, it can also be working as this in silico simulation. So I think we will see a huge boom in artificial intelligence also. And like I told you, we are actually developing a prediction model based on artificial intelligence for the therapies to see what are the best therapies to target tumors, what are the best characteristics, what the nanoparticles need to have to best target tumors and avoid side effects and avoid off-target effects, and so all of this. And so I think I'm all up to that.
Any comment, Anna?
Same with me, although we in our group we didn't use it many times, but we did once to when we started to use all these polysaccharides, and we needed to choose some because there were a lot in the line, and we could only perform the assays with I don't know how many we tested, like 5 or 6 in that same project of tuberculosis. And the first thing we did was with someone that was able to work with this in silico strategies, just to identify if our materials would have a better affinity with specific types of cells. So it helped, obviously, and it's the kind of collaborative work. It's always collaborative work because we cannot reach every expertise, and it's undoubtedly very useful. And I think it's kind of like João said, it will increase in the future.
Okay, thanks. So we have to finish soon because they have another session just after a quick lunch. But if I can abuse your patience, I would just like to give 1 minute to each speaker. There's a question here that I think is nice too. What do you suggest to a student that aims to do research in nanotechnologies for biotechnological applications? Do you have anything that you want to comment about this?
I think we always have things to comment because it's a typical question from the students. Typically, you are kind of lost. Some of you not, and have very specific ideas. But most of the students, they just know they want to do something, but don't know exactly how. So one of the things that I always say is that during your academic path, you need to try things. So engage with every opportunity that you find in your academic institutions. Then something that also helps is to try to have some experience out of your comfort zone, like João said so many times, because it's very important to challenge yourself. So don't think that you are doing a graduation or you're doing a master and you just do that, inside the square. Go out of the box. It's very important. Sometimes sometimes you can count on the teachers, people, the researchers, the people you contact with to give you ideas, but you are those that each individual knows exactly what it is. So my major advice is take some time to deeply think, to ask yourself, what do I want to do? And do something that you really want, because I do really like, because I cannot imagine what is for 40 years or something of our life to do something that doesn't realize us. It's very good to do something you really like and to wake up every day like, yay, another day.
Okay, João, you have 30 seconds.
I completely, I just want to tell you one thing, is that besides all of this, is that dreams sometimes are hard to follow, but don't let anyone destroy them. This is really important because there are people that do that, believe me. So it's it's really important that you follow your dreams. And then another thing that is really important is that you learn from the best also, and also then you teach what you learn. Because if you want to master something, you need to teach it. I think these are the two things that I like to say.
So with these wise words, I would just like that we all thank our speakers for all their contribution today. Thank you very much.
So we have reached the end of this interesting session. I want to thank once again our speakers for your willingness to be with us today and share a little bit of your knowledge.